Hello everyone, myself Animikha. I am affiliated to SRM Institute of Science and Technology, Chennai. My key research areas involve learning and memory, neurodegeneration and dementia. I'm also interested in vision neuroscience and electrophysiology. Today, I'm going to present my project work entitled Transcriptomic Analysis of Hippocampal Circle CA1 in Alzheimer's disease. Before proceeding, I would love to give an overview of Alzheimer's disease. It is a neurodegenerative disorder affecting mainly the elder population. There are several hypotheses behind this disease. One of them are tau hypothesis, another is beta amyloid hypothesis. AD results in several cognitive declines such as memory loss, loss of vision, lack of spatial and temporal senses, brain atrophy, neuroinflammation, mitochondrial dysfunction and oxidative stress, and disruption of calcium dipositive plus homeostasis. Other neurodegenerative disorders include Parkinson's disease, Huntington's disease, Lewy body disease, amyotrophic lateral sclerosis, Friedreich ataxia, etc. Three single gene mutations associated with ATR, amyloid precursor protein on chromosome 21, present in 1 on chromatin 14, present in 2 on chromosome 1. Hippocampus is known as the center for learning and memory, which is very affected in Alzheimer's disease. Here in our project, the focus is hippocampal subfield CA1. So, so here is an overview what we have done in the project work. After the labeling of data, we perform quantile normalization and principal component analysis. So here is our result. We can see the plot of principal component 1 versus principal component 2, principal component 1 versus principal component 3, principal component 2 versus principal component 3. The principal component 1 accounts for 23.01% variation. Principal component 2 accounts for 18.19% variation. Principal component 3 accounts for 14.75% variation. Based on these results, we detected this particular sample as an outlier. From our data, we get to know this sample is basically X119, which is an AD sample. We decided to remove this outlier. So, so here are the results of our Jensen enrichment analysis. So we can see this is a volcano plot. Here, the x-axis represents log to fold chain and the y-axis is the p-adjusted value in logarithmic format. So red dots here are the significant genes, where the blue dot shows the non-significant ones. The right side showed positive upregulation of the genes and the left side shows negative upregulation of certain genes. The most significantly upregulated genes are mentioned here. Here is another plot. It is gene ontology enriched pathways. So as we can see here, there are activated pathways and suppressed pathways. This the size of the circle represents the overall gene count. At the bottom of these boxes, the gene ratio is mentioned. The most significant, statistically significant ones are the red ones, while the least statistically significant ones are the blue ones. Here, in this plot, all samples are statistically significant. So here is a CNET plot of the gene networks. Here we can see the color of the node represents the fold change and the size of the node represents the size of the genes. So here are the genes and here are the some down-regulated gene ontology terms. Here fold change is negative. So it is a result of cage-enriched pathway. Again, here are activated pathways and here are the suppressed pathways. The gene count is represented by the size of the circle, and here is the we can see the red signifies the statistically significant ones, and the blue here represents the statistically non-significant ones, and the range of the p adjusted value here is from 0 to 0.1. We can see here both activated and the suppressed cage enriched pathways. Here is another Venn diagram between the 226 genes of supplementary table S5, and here is 181 genes that is the statistically significant genes from our output file after gene set enrichment analysis and sorting only the statistically significant one. We found 0 overlapping genes, and we conclude that it might be due to the fact our project is very much focused on the hippocampal surface CA1, whereas the main study took like considered every subfield of the hippocampus. We'd love to conclude our project with the fact that we have done both the gene ontology analysis and cage analysis. Geoterms such as neuronal actin potential, transmitter gated channel activity, neurotransmitter signal secretion, neuropeptide signaling pathway are upregulated. Another notable mention is olfactory receptor activity and detection of chemical stimulus involved in sensory perception. Hippocampus is widely known as for its role in learning and memory. These activated geos with respect to subfield CA1 could give us new directions for future research. The same kind of analysis could be conducted for other subfields of hippocampus as well. Limitations of the study is lack of validation dataset. Therefore, we wish to do further analysis if possible. I take immense pleasure in the successful completion of my project entitled Transcriptomic Analysis of Hippocampal Surface CA1 in Alzheimer's disease. I would love to acknowledge my research mentor, Dr. Raghavendra Lakshminaran and Dr. Elia Brodsky. I would also love to express my gratitude to other members of the team, including but not limited to Ms. Animikha Rai, Ms. Vaishnav, and Dr. Mohit Pujurkar. I am indebted to all of them. Without their constant support and motivation, this project would never have been possible. Thank you all. Thank you for listening to me.